The Synaptic and Network Development Group presents MEA-NAP, a flexible network analysis pipeline for neuronal 2D and 3D organoid multielectrode recordings.
Hey, are you recording MEA data from neuronal cultures?
Yes, right now I'm looking at the action potentials from these iPSC-derived cortical neuronal cultures. And here, if I look, I'm looking at the firing rates and the burst rates.
Did you know that MEA data reveals the microscale functional networks from neuronal cultures?
Whoa, that's amazing. That could really advance our understanding of the brain. I really want to look at these functional networks in these recordings, but I'm feeling really tired. If only there was a diagnostic tool that would look for network activity in my MEA data while I take a nap.
We created MEA-NAP, the microelectrode array network analysis pipeline. This enables neurophysiologists collecting MEA data to explore microscale network features in their cultures. In addition to detecting spikes and characterizing burst activity, our pipeline allows the user to infer connectivity and construct a snapshot of the functional network. But it doesn't stop there. Network features and properties can be extracted and compared across ages or groups, and this allows direct insight into similarities or divergences over time or between cultures for these functional microscale networks.
We have applied MEA-NAP to investigate developmental changes in human cerebral organoids. To do this, we had to develop new ways to detect spikes in these cerebral organoids, including a new template-based spike detection method. This will allow us not only to understand whether there are changes in firing rate over development, but also to be more precise to detect network changes.
We grew human iPSC-derived neuronal cultures, and now I'm going to look at them. Then we recorded the electrical activity.
Using MEA-NAP, we see the developmental increase in functional connectivity. The network density increases, for example, while the number of modules decreases.
Integrated into MEA-NAP is the mapping of roles for nodes in the functional network. We call this node cartography. It allows us to understand how integrated or how isolated individual nodes are, but also whether or not these nodes play a hub function. Using this cartography analysis, the user can generate a visual and intuitive map of the distribution of functional node roles in the microscale network.
So in MEA-NAP, we have several network dynamics features, including the effective rank, which quantifies the dimensionality of firing rates in the network of neurons, but also we use non-negative matrix factorization to disentangle different components of network activity.
And what can this tell us about the networks within our culture?
This can tell us whether networks develop sub-patterns of activity for development within different modules of nodes. This is important for understanding information processing because the number of dimensions that the network activity has can both control how much information it can contain, but also how robustly they can encode different forms of information.
I study microscale networks.
The way neurons actually talk to each other is the basis of neural computation and thought. And so to be able to actually measure and trace neural connectivity enables the study of the underlying mechanisms of those processes. And so this method enables us to actually measure not just the structure, but the functional connectivity over time across different disease-like states and across different species. And so in that way, it's opening up an understanding of not just what changes happen on a superficial level, but even the underlying functional changes that might be responsible for behavioral or cognitive differences.
Thank you for watching our video abstract. We hope you enjoyed it. You can download MEA-NAP and try it out yourself at this address.